Alright, so herpes viruses are enveloped and they're really fragile. They need to be spread with direct contact except for chickenpox, and they're really susceptible to drying out, to desiccation, to soaps. Right, this envelope is not very resistant to the environment. They are double-stranded DNA viruses. They have a pretty large genome, right? We've looked at some other viruses this semester that might only have 5 genes or only have 2 genes in the case of uh, hepatitis D, right? These have got more than 30 genes. Inside the envelope, you've got an icosahedral capsid, and the capsid plus the envelope is a relatively large virus too. And they can be pleomorphic, so the envelope is slightly squishy. They can be between 150 and 300 nanometers. And if you look at the structure of the virus, right, you've got your outer your membrane or your envelope, right? And this is going to have the glycoprotein spikes that are going to be responsible for binding and entry, and tegument, right? So that's kind of like the matrix protein, and you've got lots of actual different tegument proteins here that are responsible for some roles very early in the infectious process, for antagonizing the innate immune system and for helping the virus traffic to where it needs to go. Then you've got your nucleocapsid, which is going to protect the genome, and the genome would be inside the core nucleocapsid. So how do herpes viruses enter the cell? Well, in this illustration here, walking through it, we've got several different glycoproteins, and these are GB, GC, GD, GH, GL. So that's a glycoprotein B, D, C, H, and L. They're going to bind to multiple receptors and co-receptors at the surface. Attachment factors include the ever-present heparan sulfate proteoglycan, among others. So you get interaction with multiple receptors, and then you mediate fusion with the viral envelope and the plasma membrane. So GB in particular is going to cause fusion between the viral envelope and the plasma membrane. And you might be thinking to yourself, wait a minute, I thought they entered through endocytosis, because that's what was discussed with CMV. And CMV is a special case. So it appears that in some types of cells, CMV enters through endocytosis, and then there's a pH-dependent fusion between the viral envelope and the membrane of the endosome. And in other cell types, CMV enters through the classical herpes pathway, which is fusion at the plasma membrane. So the kind of canonical way herpes virus enters cells is we merge the viral envelope with the plasma membrane, and then the core and the tegument proteins can then enter the cell. So some tegument proteins are going to help shut down host translation, and some tegument proteins are going to help co-opt the cytoskeleton and allow trafficking of the viral nucleocapsid to the nucleus. Some tegument proteins are going to be responsible for initiating transcription. The book highlights VP16, which is tegument protein that travels to the nucleus and helps initiate transcription, and the viral host shutdown protein. So VHS, or virion host shutoff protein, and this is going to stop host translation, and that allows then for the viral proteins to be preferentially translated. And if viral host translation is shut down, that also makes it difficult for the cell to initiate an antiviral response. Now, looking at the genome of different herpes viruses, they're all going to have some things in common, but they're going to vary in length a bit. All herpes viruses are going to have some latency-associated genes. What this figure is showing you is in the boxes, the dark boxes, you've got your essential genes for DNA replication and assembly and so forth. And then in the unique regions, you're going to have genes that do all sorts of things that help to do things like determine the cellular tropism, and genes that help antagonize the immune system. So you're going to have some similar sequences to all herpes viruses, and then you're going to have some genes that are unique that are going to allow it to do its own thing, right? To replicate in different types of cells, to replicate in either fibroblast cells or epithelial cells or B cells. So DNA replication is going to take place in the nucleus. These viruses, one of the things that allows them to replicate in all sorts of different cells, including nerve cells, which tend to have pretty slow metabolisms, is these viruses encode for part of the nucleotide biosynthesis pathway. So they're not entirely dependent upon the host for making more nucleotides, your A's, T's, C's, and G's. Now, when the virus enters, it can either do a lytic infection, so be active and start producing more viruses, or latent infection. The latent infection is associated with several latency transcripts, and during a latent infection, the virus actually has gene products produced that prevent the expression of other viral proteins in the lytic cycle. So the virus will kind of hold itself in stasis until a particular stressor arises. So the double-stranded DNA genome has to circularize, and then it's transcribed by cellular RNA polymerases, and it replicates through rolling circle replication. And so again, this is better demonstrated in a video. I showed the same link in one of the previous lectures. So that the book is trying to draw a picture here. So you circularize, and then you have a nick, and then you're going to hold one kind of strand in place. And then remember, DNA replication always goes 5' to 3'. So on the 3' end of the nicked DNA, you can start adding more nucleotides to that, and it goes around in a circle, and so you kind of spin off a length of DNA here, and then that can circularize, and then you can make the other strand from that again in a circle. So take a look at the video, and that demonstrates the process a lot better than the still shots that they're trying to show you here. So the viral proteins responsible for replication, right? They're numbered in an annoying way, and only herpes virus biologists actually have all of these memorized. The UL5, UL8, UL52, and so forth and so on. But we should recognize the terms. We've got helicase primase, and those are going to do the same things that you're familiar with, right? The helicase is going to unwind things. The primase is going to lay down a primer, and then we've got the DNA polymerase, and it's using its own viral DNA polymerase. And the viral DNA polymerase, just like your DNA polymerase, needs that RNA primer to attach, to bind onto, right? So I'm not going to ask you guys to remember UL5, UL8, UL52, UL9, UL30, UL42, but you should be familiar from gen bio and molecular biology and microbiology and cell biology and genetics the purposes of the enzymes helicase, primase, and our DNA polymerase. Okay, so remember that all herpes viruses are going to share a few things in common. Number one, they can all go into latency. All herpes viruses are going to have a DNA genome that replicates the nucleus, and they can all enter latency. Now, in order to maintain themselves during latency, they might need to be growing in some cells that don't normally have very active metabolisms, as I mentioned before. So they have genes to make more nucleotides available to them. And when they are in latency, they're only going to express their latency-associated transcripts. And actually, a large percentage of the herpes virus genome is associated with latency. So HSV1 and 2, about 50% of the genome is involved in either maintaining latency or evading the host immune systems. So remember, pox viruses had those virokines. Well, herpes viruses have virokines too. So an example here is they have an IL-6 analog, which promotes cell survival, so the cell can't trigger apoptosis, and also downregulates the interferon alpha response. Now, latency also helps it hide, because when the virus is latent in your neurons, it's not producing any active virions. It's not producing any proteins that are going to be on the surface of the cell that could get tagged or taken out by cytotoxic T cells or tagged by antibodies. When the cell is latent, it's not producing virions, which means it's not really detectable by your immune system. Your immune system can really only fight off herpes virus during an active lytic infection. So after the initial infection, it's going to go dormant in your cells. Now, in this figure, it's the sensory neurons, so this could be herpes simplex virus. If this was, say, Epstein-Barr virus, then it would be B cells. So depending on the type of virus, the cells that it goes latent in is going to vary. But while it's latent, it's only expressing those latent transcripts, and the virus has different ways of maintaining latency. So one way it can do that is by stopping transcription. So it can bind to transcription factors, or it can have other proteins bind to the genome that prevents transcription. The other mechanism is using complementary sequences. These are microRNAs to silence gene expression. So the way this works is you have a sequence of RNA that is complementary to a transcript, to a messenger RNA, and the microRNA gets processed by the cell, and it gets complexed with some enzymes, and then if that matching messenger RNA makes it to the cytoplasm, then it gets degraded before it can be translated into proteins. So we've got a couple different ways that we can silence the viral genome and maintain latency. Now, some people are starting to target these latency mechanisms with antiviral drugs because if you can stop latency, that sounds kind of counterproductive, then the immune system can do a better job at fighting the infection. So when you have some sort of stress, again, physical or emotional stress, then the virus pops out of latency. This is also enhanced if your immune system is compromised. So this is why elderly patients more frequently have shingles, for examples, because your immune system is starting to wane, and then you have some sort of stress on top of that, and boom, you've got shingles. So that shingles vaccination is really important. So then the virus reactivates and it travels down the nerves to the epithelial cells where it can cause an active outbreak. Now, in these cells, and after the outbreak, your immune system does recognize it, so your secondary outbreaks are usually not as severe as the first one. So remember, your first outbreak can be between 1 and 2 weeks after your initial exposure, and then subsequent outbreaks can be weeks apart, months apart, or years apart, or in the case of chickenpox and shingles, decades apart. All right, so like some other viruses that we've talked about, herpes viruses have very regulated and ordered gene expression. They have immediate, early, and late genes. And so some of the early genes are going to help regulate and turn on the late genes, and so forth and so on. So you've got to have this ordered progression. So the virus binds to the surface of the cell, different receptors including heparan sulfate proteoglycan. The fusion occurs at the plasma membrane. Remember, sometimes CMV can enter through an alternative method, through endocytosis, and that broadens its cellular tropism. But the classic way herpes viruses enter is shown here. So then the virus traffics, the core of the virus traffics to the nucleus. Inside the nucleus, it's going to start transcription, right, of these really immediate early genes. The immediate early genes get translated, come back to the nucleus, and then they can start activating the next set of genes. Right, so then we're going to start getting synthesis of the genome. Remember, that happens for that rolling circle replication. So again, check out that that animation that I've linked to. So you get this really long string or concatemer of DNA, and then your genomes, once you've got your late genes synthesized, you've got your tegument, and you've got your capsid, you can package your genome. Now, herpes does something really interesting here. It's one of the only viruses we know of that actually buds out of the nucleus. But then it loses that first envelope, and it picks up another envelope in the Golgi. The choice between lytic and latent infection. So if we look at this figure on the right, we've got over here, if we look at this, we've got our productive or lytic infection. And so we immediately have transcription starting, and the immediate early genes can then produce the early proteins. We get our genome replication, and then we produce our late proteins, and then we can make virions. Now, in latent infection, what this is showing you here is you've got some proteins, histones or histone-like proteins, that can bind up the DNA. That's going to prevent transcription. We've also got other latency-associated transcripts that are going to degrade any messenger RNAs that manage to get transcribed with the microRNA silencing. And so this is our latent side. And then in here, if you have stress, you can get reactivation and then start your lytic infection. Okay, so the formation of the membrane. So herpes has that envelopment, de-envelopment, and then envelopment again, which is pretty cool and pretty unique. So the capsid, once you make the capsid and the DNA and everything is packaged inside, then it actually buds out of the nuclear envelope, and then it loses its envelope, and then it gets associated with more tegument proteins, and it goes through the Golgi and picks up another membrane, and then it leaves through exocytosis. And there's actually a really cool video here where they did a lot of electron microscopy, and you can actually see it here budding through the nuclear membranes. Let's see if this will play. So it picks up a membrane, and then it loses the membrane, which is kind of cool. I'll do that one more time. Right, so it picks up that membrane from the, because remember the nuclear envelope is a double membrane structure. So it buds through the first membrane into that inner membrane space, and then that membrane merges with the second membrane and it loses it. And this we think is pretty unique to herpes viruses. And then it picks up its second membrane and leaves through exocytosis. Alright, so after our productive primary infection, remember this is pretty shortly after your initial exposure, then we're going to hijack retrograde transport, and we're going to move up the axon and establish latency in the cell body of a nerve cell. So again, this is showing nerves for most herpes viruses like varicella zoster and herpes simplex 1 and 2. But if this was Epstein-Barr, for example, or human herpes virus 6 or 7, then it would be different cells. Okay, and this is just a good diagram from your book, sort of demonstrating after you experience some sort of stress. You're going to get some viruses. They're going to travel back down the axon, and they're going to get into the epithelial cells. And you've got dormant virus here, and then once it starts to reactivate, you might notice some red, starting of a rash, red rash, and then the virus travels up, and you get the vesicle formation, and then your immune system is going to fight it. It's going to blister over and go away, but the virus is still going to remain latent in your nerve cells. So remember, in true latency, there are no detectable virus particles. But if you're in that prodromal phase, when the virus is just starting to come out of latency, you're not going to have vesicles yet, you're not going to have the active outbreak yet, but at that point you can still be considered contagious. So different antiviral treatments. So acyclovir is one of the classics. It's a nucleotide analog. It's an analog of guanosine, and it can act as a chain terminator. And then all of these others, all of these other related acyclovirs. So penciclovir, right? Famiciclovir. All of these are going to work in a similar fashion. And then you have a few other medications that are targeting other proteins as well, other viral proteins as well. But there are a lot of different herpes virus treatments out there, and so while some strains may be resistant to some drugs, it's important that it's important to know that herpes can be treated. A person can have herpes virus, and if they're on treatment and it's well managed, not transmit it to their partner. So there is a vaccine for chickenpox. Generally, it's safe, it's a well-tolerated vaccine, and generally it is effective. Occasionally, you do have a person who is vaccinated and they wind up getting chickenpox later, and this occurs in a pretty small percentage of individuals, and this is usually a very mild case. Remember, someone who has full chickenpox, if they get infected naturally, can have 500 lesions, and so we're talking at like an order of magnitude less here. And the shingles vaccine, it is important to get that, and that reduces the chances of shingles significantly. And just as a reminder, kind of reaching back to the oncolytic chapter, some herpes viruses now are being used as therapeutic agents in treating cancers. So this is a pretty cool technology that they're starting modifying these herpes viruses to be therapeutic and to treat brain tumors. All right, we're going to end here. This is going to end the herpes chapter lectures. Hope you guys are doing well.